Hello, everyone. Welcome to today's live broadcast, UHPLC and HPLC Method Development for Pharmaceutical-Related Substances, How to Translate Methods, Maximize Resolution, and Comply with USP Chapter 621. I'm Laura Bush, the Editorial Director of LCGC North America, and I'll be your moderator for today's event. We are delighted to bring you this educational web seminar presented by LCGC and sponsored by MacMod. MacMod's mission statement is Smarter Chromatography. But you may ask, what does Smarter Chromatography mean? It means that when they make a recommendation to you about an HPLC or UHPLC product, it is to partner with you to offer innovative solutions to your complex problems. They use advanced, state-of-the-art analytical software to match their product portfolio with your separation needs. They leverage their company's 29 years of experience in the LC column business, along with their technical expertise and manufacturing network, to help you solve your toughest application problems. MacMod provides up-to-date and accurate technical catalogs and reports from industry-leading separation scientists to keep you informed of new technologies. That is what Smarter Chromatography means. We have a few housekeeping announcements before we get started. The webcast is designed to be interactive, so we encourage you to ask questions during the event. You can submit your questions at any time by typing them in the Q&A box, and you can find that by clicking on the red Q&A widget at the bottom of the presentation window. You can enlarge the slide window by clicking on the small green icon in the upper right-hand corner of the slide window, or by hovering your mouse over the lower right-hand corner and dragging the window to the desired size. The slides will advance automatically during the web seminar. And if you have any technical problems viewing or hearing the presentation, please click on the question mark Help widget in the dock at the bottom of your window. I would now like to introduce today's speaker. We are very pleased to be joined today by Alan P. McEwen. Alan McEwen completed his PhD in analytical chemistry at the University of Nottingham in the UK. His postdoctoral studies focused on the development of novel stationary phases. Alan has held individual contributor and senior analytical leader roles in the pharmaceutical industry at AstraZeneca and Pfizer across a 13-year period. In 2011, Alan joined Advanced Chromatography Technologies as the Business Development Director. Alan's role is both commercial and technical and includes R&D, new product design, sales, marketing, and technical advice. Alan also works directly with customers for chromatography support, troubleshooting, and method development. He has published various peer-reviewed articles and written several book chapters on separation science, chromatography, and genotoxic impurities. And he is a Fellow of the Royal Society of Chemistry. Alan, thank you for joining us today. You now have the floor.
Thank you, Laura, for that very kind introduction, and welcome to everyone who has logged on to listen to this webinar. My name is Alan, and hopefully over the next 45 minutes or so, I'll share with you various different elements of method development, method translation, and the power of selectivity. As a brief outline to this webinar, I'll start by reminding us what selectivity is. I'll then talk a bit about how we can use selectivity to optimize our method development approach by considering key variables available to us to change to develop our method. I'll then use this as an example and develop a method, a related substance method, something that we've done recently in our laboratory. I'll then move on to method translations, and I'll discuss isocratic method translations between different particle platforms, porous particles and solid core particle platforms, different particle sizes, and also different instrumentation. And that will be with regards to isocratic and gradient separations. And then finally, I'll finish the seminar by discussing how you can maximize resolution for complex samples, which is one element of method translation. So first of all, what is selectivity? Selectivity is a central and key concept in chromatography, and it's described very simply on the screen at the moment. So the selectivity factor or separation factor can be thought of as peak spacing. It is the ratio of retention factors between two adjacent peaks. Why is selectivity so important in chromatography? Well, it helps us with resolution. Resolving compounds of interest is what we do as chromatographers. And as you can see from the graph on the slide now, you can see that alpha or selectivity is the key to resolution. Now, of course, efficiency and retention factor are also important in chromatography, but in terms of the power of resolution, alpha is the key. Now, there are lots of factors that affect selectivity in chromatography. And from a practical perspective, on the slide now we have isocratic separations on the left and different variables, mobile phase variables, available to you to change to affect selectivity. All of these parameters have an influence on selectivity. However, they are in a list format, so in a bulleted list format. So the parameters at the top of that list have a bigger influence on selectivity. So the type of stationary phase, the eluent pH, and the type of organic modifier are very powerful parameters in method development. And of course, for gradient separations, these parameters are important too, but we have some other parameters we need to consider, such as the gradient steepness, we have a gradient time, and column dimensions that we also have to consider. Now, selectivity is integral to method development. Whenever we change parameters in our method development activities, it has an influence on selectivity. Now, there are lots of ways to do method development, which is great because that makes it exciting for us as chromatographers. But on the screen in front of you is one possible approach to method development. In the box on the far left-hand side, you can see is the simplest approach to method development. We have one instrument which has one column, operating at a single temperature with one mobile phase eluent, normally acidic pH with an organic modifier, acetonitrile or methanol are the most popular, and then one scouting gradient run. That is the simplest approach to method development, performed every day all around the world. Now, of course, we can make that approach more complex if we do not get the separation that we're looking for. We can consider other variables, such as in the second box, we can look at low and high temperature. How does this affect my separation? How does it affect the resolution of our compounds? We can look at the type of organic modifier. Methanol and acetonitrile, as I've already mentioned, are two of the more popular solvents. We can look at column stationary phase, the type of bonded phase that you use. And I've bunched together three different types of stationary phase there, which are common in liquid chromatography nowadays. So we've got bonded phases based on alkyl chains, such as super C18, the C18 and C8 type phases. These are dominated by hydrophobic interactions between the analyte and the stationary phase. We have aromatic stationary phases. These are phases that contain a aromatic delocalized ring system, the simplest of which is the phenyl type stationary phase, but there are alkyl phenols and even pentafluorophenyl type stationary phases. And then we have phases that allow some type of polar interaction. The pentafluorophenyl will give us some dipole type interactions as a mechanism of interaction. We've got polar embedded phases such as the C18 amide, and then we also have a cyano phase there, giving us a slightly different selectivity again. All of these different types of phases have different mechanisms of interaction, and they affect selectivity in a different way. That's useful for us in method development. And then finally, the last box on this continuum we have is mobile phase or eluent pH. pH can be a very powerful parameter in method development. When you change the ionization state of your analytes and they gain or lose protons, it can have a huge effect on retention in reverse phase chromatography. Now, as we move from the left-hand side of the slide to the right-hand side of the slide, we do more injections, we generate more data, it takes more time, but we generate more knowledge as well. Now, rarely do we have the opportunity to explore all of these parameters to get a complete understanding of the retention behavior of our analytes in the mixtures, and so we must look to rationalize those parameters accordingly. So, of course, the question is, which parameters should we focus our efforts upon? Now, in today's talk, I'm going to focus on the power of the stationary phase or the bonded phase type. I want to demonstrate how understanding the different mechanisms of interaction between the bonded phase and the analytes can be very powerful in terms of maximizing resolution via selectivity. So here we have an example of a degraded sample, and this is analysed on a standard C18 column. This is dominated by hydrophobic interactions, and we can see, because it's an unknown, as a degraded sample, it has an unknown number of peaks. So how do we know how many peaks are in this sample if we were wanting to characterise this sample to understand its composition? Now, as you can see, we've got a general scouting gradient run there, UHPLC type separation, in around 5 or 6 minutes. And if you count the tops of the peaks, it's around about 22 peaks. Now, there are lots of options for us now to modify this chromatography to see if we can see if there are other components in this sample. But what I'm going to do is simply keep the method conditions the same, the gradient the same, the mobile phase, the pH, everything, and the sample the same, but I'm going to take the C18 column out and put a different stationary phase in. And the stationary phase that I'm going to put in has different mechanisms of interaction. The C18 PFP has a hydrophobic interaction element, but it also has pi system interactions due to the delocalized ring. It has a moderate dipole moment interaction due to the fluorines that are substituted 5 times onto the aromatic ring, and it also has a degree of shape selectivity. So we're looking at different mechanisms of interaction with this column compared to a standard C18. So if we analyze the same sample under the same conditions, but just with this different stationary phase, we now see that there are 28 peaks in this sample. So we've moved the peaks around in the chromatographic space. We've exploited the selectivity of the different bonded phase to help us understand the composition of this sample. And now what we need to do is map the peaks between the different chromatograms to help us understand where analytes have moved around in the chromatographic space. Now, in this example, this is a simple example using LC-UV. Most powerfully, you would use LC-UV-MS, so you could use the power of the mass spec to track those peaks as they move around the chromatogram too, giving you that extra information as well. And what I'm showing on the screen is a very simple screening approach, and sometimes you find these set up in laboratories in all around the world where people have 2 or 3 or 4 different columns with different selectivities or bonded phases, and it helps them understand different elution orders, different compositions of the samples. It helps them understand what is in these samples. And then by peak tracking, hopefully we can see everything that's in that sample. So stationary phase selectivity is very powerful. We're using the power of selectivity through the stationary phase. As another example, we have here a solid core phase. Now this is an ultra core 2.5 micron super C18, and this is a separation of water soluble vitamins. And we can see under this very general sort of gradient conditions that there are areas of coelution. But by switching the bonded phase to a super phenylhexyl based solid core technology, we get a different elution order and complete separation of these compounds in a relatively short time frame. So again, we're using the power of the stationary phase to give us the selectivity. We understand the selectivity differences between a C18 and a super phenylhexyl, and it's giving us this separation. So I've talked a little bit about selectivity and different columns. How do we know if columns are the same or if they're different with regards to their selectivity. And of course it's dependent upon your application with your particular analytes. And the approach on the screen is something that was published back in 2006, and the reference is at the bottom of the slide. And this is a very nice approach to understanding the selectivity differences between different types of columns. And what this approach discusses is that you take a column 1, which is normally a C18 for most people, and you run down this column different analytes of interest with a certain method of your choice, and you note the retention times. You then very simply take that first column out, put a second column in, and you use the same method and the same sample and the same analytes, and you very simply plot the retention times against each other. This gives you a regression coefficient, a line of best fit between the two columns, which you plug into the equation, the selectivity equation, and it gives you a selectivity value. And essentially selectivity values from about 8 upwards, you deal in whole numbers, whole integers for this type of approach, means that there are suitable differences between the two columns with that particular application or analyte class, that you're going to move peaks around in the chromatographic space. So this means it would be helpful for method development. Now of course you can get larger selectivity values too, so you're getting much more differences between the two columns in terms of selectivity. And if you change multiple variables, then you can get even larger values too, meaning you're really moving peaks around in the chromatographic space. So this is an approach that helps you understand the screening approach of different columns with the same mixtures under one or two different conditions. So what does that look like when we look at lots of different analytes? Well, what we have here is an ultra-core, a solid core based example on a super C18 versus a super phenylhexyl at low pH. And we can see here with about 50 odd analytes, small molecules, different molecular weights, different log P's, we've worked out the regression coefficient and calculated the selectivity value, and it gives you a value of 19. So you can see there's some scatter in this graph. So we're moving the peaks around in the chromatographic space. They're changing elution order and changing retention time. Now if we look at what that looks like for a whole host of different analytes, we can see here a 23 component mixture, and we can see that we're affecting the peak spacing, we're affecting the elution order, so we really are moving those peaks around in the chromatographic space. Now, I've talked about bonded phase selectivity, but also solvent is always a parameter that I look to use in my method development activities. Lots of people use acetonitrile as their first solvent because it gives us some beneficial advantages, such as a low viscosity for our separations, which keeps pressure low. However, using methanol is also helpful. Now, methanol, if you're a mass spec person, is known as a helpful solvent because it can help you with ionization and detection and sensitivity of your analytes with the mass spec detector. Now, what we have on the screen here is a simple acidic separation, acidic mobile phase based separation of 18 components using a C18AR. So this is an alkyl phenyl type stationary phase. By switching the solvent from acetonitrile to methanol, we affect the elution order and the selectivity of this particular separation. And actually, if we calculate the selectivity value, it's a value of around about 15 if we were to do this in a graphical format. So again, what we're seeing is a change in the elution order, a change in the selectivity between the same bonded phase but two different solvents, between acetonitrile and methanol. It's always worth considering the solvent type in your method development activities. It helps you explore the selectivity space. So if we calculate different selectivity values, so this is now in the triangle I'm showing you is a methanol-based mobile phase. There are three stationary phases on the screen, C18, C18AR, and C18PFP. And on the right-hand side of the slide are the main mechanisms of interaction with these three different phases. Now, if we calculate the selectivity values using the graphical format that I showed you a few slides back, then we can calculate the selectivity values to be 10, 11, and 12, which are reasonable selectivity values showing that with these three phases you're moving the peaks around in the selectivity space. Now we can repeat that work and actually also do it in acetonitrile, and we can see here the values are a bit smaller. The values we now have are 7, 8 and 8, and that's because it is known that acetonitrile suppresses pi-pi system interactions. So another reason to explore methanol for aromatic-based stationary phases is it helps with pi system interactions between the analyte and the stationary phase. And we can also plot the data between the different stationary phases as well, between the different solvent planes as well. So here we're using a single stationary phase, which is C18, and we're plotting the changes in selectivity from a C18 in acetonitrile to a C18 in methanol. And we can see we get a selectivity value of 15. Now, acetonitrile is an aprotic solvent and methanol is a protic solvent, so they're affecting the selectivity, the elution order of the analytes in these particular mixtures. And we can calculate the data between the two different solvent planes, and we can see the values 15, 15 and 18. And then, of course, because we're using Excel for this type of data analysis, we can plot the retentions times on a C18 in acetonitrile versus a C18 PFP in methanol, and we can see we get a selectivity value of 19, and so on and so forth, calculating these different combinations and permutations of solvents and three different stationary phases. So what does all this mean? Well, what I'm showing you on the screen now is a pragmatic method development platform. This is based on real retention data with a whole host of different small molecule analytes. And what I'm showing you is that using these three different phases with two different solvents, we are fully exploring the selectivity space. We've rationally chosen those different bonded phases together with methanol and acetonitrile to give us the different selectivities that we can see on the screen now. Now, of course, you can make this as complex as you like. If you wanted to add another column to this mix, to this three-column method development platform, you would draw a square and calculate the different selectivity values in methanol and different selectivity values in acetonitrile. But as ever with these things, how many variables do you explore and how much time do you have for your method development? This is a three-column, two-solvent method development platform based on real selectivity values, which tells us that we will move peaks around in the selectivity space. Now, I've shown you those three different columns in a triangle format, and here are three different columns again with different mechanisms of interaction, but now I'm showing you chromatographic data on a simple 7-component mixture. So here we have three different columns that would be maybe more suitable for more polar type species. We have a cyano-ES based column which has a different mechanism of interaction and a different elution order of the compounds compared to a polar embedded C18 amide which has a different mechanism of interaction and different elution order compared to a C18 PFP which has a different mechanism of interaction and a different elution order. So using selectivity of the bonded phases, this is all under the same conditions, same mobile phase conditions, but moving the peaks around in the chromatographic space. So depending on your analytes and the type of mixtures you deal with, you can choose 2 or 3 columns of your choice which best describe the types of analytes and applications you have, and you can fully explore selectivity. But the important thing to note is that you've chosen those different columns with different mechanisms of interaction because you know they give you different selectivity values. So based on the workflow that I showed you towards the beginning of this seminar, I showed you 5 boxes. What I've now done is shrunk those 5 boxes down to 2. So we have 3 columns, 2 solvents, but we've still got an information-rich data process that should allow us to fully explore selectivity. So to summarize the first section of what I've talked about, hopefully what you've seen is that selectivity is the key to resolution. Alpha really is powerful with regards to method development and resolution of compounds. And I've shown you that many different method, mobile phase in particular, and instrument parameters can affect selectivity. And if we understand which ones are more powerful than others, we can focus on those key variables that are going to give us more information for the injections that we do. You can optimize your workflows, as I've shown, where you can reduce multiple different parameters down to far less parameters, but hopefully retaining key retention information. And finally, based on these data that I've showed you here, I showed you a 3-column, 2-solvent optimized method development platform, which would give you information on your analytes, but it's all based on selectivity, which is the key to resolution. So that finishes the first section of this talk, and now I'm going to show an example of a method development activity using this stationary phase-based screening approach. So how do you do method development? On the screen at the moment is one way to do method development in 4 easy steps. Most people start with some general scouting runs. They then optimize their chromatography based on their data from step 1. They may be optimizing for peak shape or run time and so on and so forth. They then validate according to their local SOPs or guidance. If it's the pharma industry, they will take information from the ICH time guidelines. And then finally they implement their new method and begin analyzing samples, or maybe depending on the company or department they work in, they transfer that method somewhere else and somebody else does the sample analysis. So I showed you a short 3-column 2-solvent system, and I'm going to use that as the basis for this example. So first of all, lots of people would generally perform a broad scouting gradient run at an acidic pH. This is what I always recommend. How do you calculate your starting conditions? Well, it depends on the columns you have available to you. For gradient chromatography, their dimensions, and from that you can calculate your flow rate, and of course depending on the type of system you have, what your maximum pressure is. And we know from the work of Dolan and Schneider and so on that they generally recommend a K-star retention factor in gradient chromatography of greater than 2, but less than 20 for your initial method development. And you can see, based on the example I'm going to show you, we're using 100 by 3 millimeter base columns, 1.2 mils per minute flow rate, and if you work out the various different parameters and so on, you can see that gives us a K of around about 3 for our analyte soluting. Now the example I'm going to show is based on acetaminophen and its impurities, or some of their impurities. Now on the screen we have 10 related molecules to acetaminophen, including acetaminophen itself, and the data, the physicochemical data I've also put on the screen. You can see there's the pKa values, there's the log P value, a measure of lipophilicity, and I've also calculated log D values at specific pHs to give us an idea of the solubility of these compounds at within aqueous and organic environments. This is the initial screening, so this is similar to the triangle that I showed you, and you can see that the screening was done at pH 6, and the reason for that is this first eluting compound, 4-aminophenol. You can see that it's highly water soluble, very aqueous water soluble, and so using in reverse phase chromatography, it's important that we get this compound retained. So you can see the log D value at 5.5, pH 5.5 was negative 0.04, so we chose a pH value of 6 to try and get some retention of this particular impurity. Now, as we look down the slide, you can see we have the C18 on the top chromatogram, the C18AR in the middle chromatogram, and the C18PFP at the bottom. And we can see differences in the chromatography. In the green box, we can see peaks co-eluting and actually switching around between the different phases. The red bar shows you differences in the retention time or resolution between peak pairs and the peak pair 6 and 7, and then the purple box also shows you some co-elution and some separation as well. Now, based on this initial screening data, we decided to choose the C18PFP for further work. Now at this stage we're fortunate enough to have modelling software available to us, so we can do some modelling, but actually the separation given by the general screen on the C18PFB is not bad at all. As you can see, we've just about got separation more or less of most of those compounds. It wouldn't take much for us to optimize that to get full separation, but I'm going to move this into a modelling situation. And for the input runs for modelling, this is a gradient time versus temperature modelling platform that I'm showing you. So here are the 5 different input runs that we used, and we plug those into the software, and we produce a resolution map, which is on the screen now. And the resolution map, the color red in the fancy colored graphic, means that you've got separation of your critical pair, and as it goes through orange, yellow, green to blue, getting colder, you've got less separation or no separation of your identified peak pair. So you can see what I've done here is I've taken the chromatogram A. These are predicted chromatograms now from the software. From the blue section I've taken a chromatogram, and you can see we've got complete correlation of the 2 compounds, peaks at the end of the chromatogram. For B, in the yellow color, you can see we've got partial separation of the 2 compounds, and chromatogram C, the predicted chromatogram, within the red area, we've got full separation of the 2 components. So if we look at the predicted separation, this tells us that everything should be separated in our chromatogram, and we compare that to the actual chromatogram, so we actually run this on the instrument based on the conditions that we get. You can see we get pretty good correlation between the retention times of the predicted separation versus the actual. And of course, with modelling software, the benefit is that you can try and push the chromatography as hard as possible. And here we can see we've included a step gradient now in our chromatography, and between the predicted and actual, got reasonably good correlation between the predicted and experimental data. But as we're now running quite a rapid gradient, quite a fast analysis time, less than 4 minutes separation for all of these compounds, we start to see a little bit of movement in the actual correlations between the predicted and experimental data. But showing you the power of the software, it is quite powerful to get separations quite quickly and achieve your goal of resolving all those compounds in that mixture. So that's the second section of this talk. I'm now going to move on to method translations. So I talked a bit about selectivity, I've talked about bonded phase and how we can apply that in method development, and I've shown an example of acetaminophen and a few of its impurities, and how screening through different phases sometimes you can happen upon the conditions that give you full separation. And now that you've got that separation, I'm going to talk about how you can translate that separation between different particle platforms to speed it up or to improve resolution, and all of those different things. So what are you trying to achieve with your method translations? Do you want the same separation, you just want it faster? Do you want to go into the realms from converting from HPLC to UHPLC, but you want to maintain the quality of the separation in terms of efficiency, selectivity, or resolution? How do you do that? Do you want to convert from UHPLC to HPLC methods? So maybe you have a UHPLC method development platform which gives you rapid separations and multiple variables screening system, which is fantastic. This is the beauty of UHPLC. But maybe you want to give that method to a laboratory that doesn't have UHPLC. How do you translate that method whilst maintaining separation? Do you want to change the particle platform? Do you want to change from a porous particle to a solid core particle? How do you do that? What are the things you need to think about? Do you want to do faster method development? So you can get what used to take us a week, 6 or 7 different variables by HPLC. Now we can do that within a working day. Is that your goal? Or do you want to improve the resolution of your separations? Is this for a related substances compound, related substances activity, or is this for complex samples such as natural products or biomolecules or semi-synthetics? All of these topics fall under the method translation activity. Now I'm not going to cover all of these today, but I think I cover most of them. So I'll go through the examples, and hopefully there'll be something of interest for everyone in that. So first of all, I'm going to talk about isocratic method translations, and of course this brings in the USP 621 chapter, the general chapter on chromatography, which was updated in August 2014. This was a very significant update, and it is very much recommended that you read the new text for that chapter. There is no way that I can do it justice in this webinar. I've highlighted some of the key points in this webinar, but you really must read that chapter itself. I know a lot of work went into writing that chapter with a lot of discussion, and I think it's an excellent update to the original isocratic recommendations. So isocratic LC methods can now be converting using the column length to particle size ratio. And in fact, we can use minus 25% to plus 50% of the original L over DP ratio specified in the monograph. Also, column length to particle size ratios outside this range can be used if you want to use the peak efficiency option. So this new rewrite of USP 61 has incorporated a significant degree of flexibility for those analysts looking to convert traditional, older pharmacopoeia-based methods, isocratic methods, to newer technologies such as solid core technology, or to shorter column formats with smaller particles. However, there have been very little changes, if any, for gradient monograph methods. So what I'm going to discuss here only applies in terms of the USP guidance and the monograph guidance to isocratic methods. A key point to remember for monograph translations is when you convert to smaller column dimensions, it's so important that you think about the extra column band broadening effects of your instrumentation. Just because you can convert to smaller column dimensions and therefore smaller column volumes doesn't mean that you should, because the dispersion effects from your instrument can very quickly destroy your chromatography, leading to poor peak shape and very broad peaks. So you need to understand whether you're working with HPLC or UHPLC and the dispersion of your system in relation to the short column dimensions that you want to move to. So the USP guidance talks about maintaining a constant column length to particle size ratio, and this is very nicely discussed in the paper cited at the bottom of the slide, which was a stimulus article to the change in the USP. By keeping the constant column length to particle size ratio, the L over DP value, you should get similar performance with respect to efficiency, according to chromatographic theory. Selectivity and resolution should be similar if the particles that you're using are scalable. Sometimes you can get changes in alpha, selectivity and resolution, RS, depending on if you're moving between different column vendors, different platform silicas. So as an example, in a monograph that specifies a 300 mm by 3.9 mm column dimensions, 10 micron particle size, it's approximately equivalent to a 150 by 4.6 mm 5 micron column. The value is 30,000 for the L over DP ratio. And if we look at the grid for L over DP ratios, I've constructed this grid here where we've got column lengths along the top and we've got particle size going down the side. And you can see I've calculated the different values and I've highlighted there 30,000. So you can see the 300 by 10 micron particle combination. And then we can see the 150 by 4.65, well, 150 and 5 micron column combination and the 75 by 2.5 column combination. So if we look at an example monograph, and here we have the monograph for estradiol, within the USP. And here you can see this is simple separation. It uses a 10 micron L1 phase, 300 by 3.9 mm, length to particle size ratio of 30,000. And that is the separation that we would typically get. Now, if we wanted to improve the chromatography and use a smaller column dimension, then what we have here, you can see our three easy steps. We find a suitable new column, such as the 150 by 4.65 micron. We scale the flow rate according to this equation here, and we scale the injection volume to give similar sensitivity using that equation there. And you can see I've calculated those values out, and when we use that column with those new conditions, this is what we get. So we get a faster separation using the same L over DP ratio column, but this is now using a 150 by 4.6 millimeter 5 micron column. And within the USP monograph, we can see that the system suitability criteria in the little table, a resolution value of greater than 2 between estrone and estradiol, and percent RSD of less than 2, which was achieved with this new column. So a very simple translation for this isocratic method. Now, of course, we can go even faster if we wish, and the USP guidance provides us with flexibility. So as I mentioned at the beginning of this section, the USP guidance allows us to go minus 25% to plus 50% of the L over DP ratio of the actual monograph. So in the blue box there, you can see L over DP is equal to 30,000, and if we calculate minus 25% of that and plus 50% of that, we can see that we're allowed to actually choose a column length to particle size combination that falls between 22,500 and 45,000. So if we look at our column grid again, we can now see a lot more options available to us to convert our separation. We would still be complying with the guidance if we chose columns within those red boxes. So whereas previously we had 3 red boxes, now we have much larger options. And we can use that, and we can go much faster with our chromatography. And now you can see here with the top chromatogram is the original separation, and then the bottom chromatogram is a 50 by 3 millimeter 2 micron particle, and you can see within the table it still meets the system suitability criteria, but now the runtime is 2 or 3 minutes compared to 8 or 9 minutes originally. And of course we can convert to solid core technology as well, using the flexibility of that length to particle size ratio range. So at the top chromatogram we have the original separation. At the bottom chromatogram we have a 100 by 4.6 millimeter 2.5 micron solid core particle, and we can see this gives us a length over particle size ratio of 40,000, and you can see nice sharp peaks there, a rapid analysis within 3 to 4 minutes, still meeting the system suitability criteria. Now, the important thing to remember about solid core technology, and I'll discuss this more in the gradient section, is that you have to account for the differences in the particle porosities. So the solid core particles have different porosity compared to porous particles. So when you're calculating column volume, or VM, it's important you take account of that. And of course, as I said at the beginning, it's really important that we look at the negative effects of extra column band broadening on our chromatography. When you're using small columns with small dimensions on systems that have a larger dwell volume or a larger extra column volume, then there can be negative effects due to band broadening. So, and this is another example of an isocratic method translation, and this is for aspirin and some related substances. So we can see aspirin, or acetylsalicylic acid, is the main component, and we can see impurities spiked in at around about 0.5%. Now here is the separation on a porous particle platform, 150 by 4.6 millimeter 5 micron. This is isocratic translations. So we're looking to convert the account for the column volumes, scale the injection volume to give us a similar separation. And we can see here, when we go from the porous particle on the top, on standard Agilent 1100 HPLC, to the ultra-core in the bottom separation, we can see we get pretty good conversion. We get a faster separation, and this is due to the reduced hydrophobicity of the solid core particles. And of course, we ask the question, this is now a sub 10-minute separation compared to 25-minute separation. Can we go faster? Of course we can. We can translate the method to a shorter column dimensions. However, we hit a problem. So now we can see we start to lose some of the quality of the separation. We can see that the resolution between the first and second compound has started to go down on this Agilent 1100 system using a 75 by 3 millimeter column, 2.5 micron particle size. So we've lost efficiency and lost resolution, and this is due to the impact of extra column band broadening. So how can we fix this? Well, one option we have to fix this, if we can't change instrumentation, is that we can increase the column volume. So we could move from a 75 by 3 millimeter column to a 75 by 4.6 millimeter column. And in fact, when we do that, what we see is due to the increase in column volume, we now get back the quality of the separation. So we're still on the Agilent system, but the increased column volume now gives us back the efficiency and it gives us back the resolution. And if we align the different chromatograms for these isocratic translations, so at the top chromatogram we have a porous particle, and then the second chromatogram is a solid core 5 micron, and 3 and 4 we have solid core 2.5 micron particles, we can see we get very good selectivity. We get very similar retention times across the different particle formats and particle platforms, showing excellent scalability between these different phases under isocratic conditions. Now, gradient chromatography is slightly different. This is a slightly more complex situation, and there are 5 steps to gradient translations which have to be accounted for, and the equations are on the screen here. Now, all of these are pretty straightforward, but the one that's perhaps missed out the most is step number 5, and this is where we have to determine whether we have an injection hold or a pre-injection to give us an accurate translation. Also, column volume can be determined experimentally, which can be quite important, and also the dwell volume of your system should be determined experimentally too, to give you the most accurate translations under gradient chromatography conditions. Now, this slide shows you when you need to pre-inject or when you need to hold your injection, depending on this difference in dwell volume to column volume ratio. Lots of instrumentation nowadays allows you to do an injector program, which allows you to actually program in when you inject, when the gradient starts, and so on and so forth. But not all instrumentation gives you that option. So if we look at some simple translations under gradient conditions now, we've got steps 1 to 5 here. We want to maintain the quality of that separation, and we can translate that to a smaller column. We're going from a 154.65 micron gradient analysis through to a 50 by 3 micron, 50 by 3 millimeter 2 micron analysis, porous to porous translation, and we can still maintain the quality of the separation, but in a third of the time. And of course, we can go much faster than that too. We can go to, we can increase the flow rate, we can change the gradient time, still got full separation, but now in less than 10 minutes. And in this example, we only need to consider steps 2, 3, and 5. So what about translating gradients from porous to solid core platforms? So here we have acetaminophen, a different separation of acetaminophen with its impurities at 0.5% under a porous separation conditions. Now we can translate that to a 5 micron solid core platform quite straightforward using those 5 different steps. And here we can see that separation, and we've used the advantage of the solid core technology to give us a faster separation. We've had to adjust, of course, for the different column volume due to the solid core particles, and therefore adjust the gradient time accordingly. But you can see that the peak capacity is good and the resolution is good. Now with this separation, in the top chromatogram is the original separation, the bottom one we're going to an even faster separation this time. And now we can see we're maintaining the quality of the data by giving full separation in less than 7 minutes, and we can do it again in less than 3.5 or 4 minutes, giving us full separation of this complex mixture. Why correct for the dwell volume to column volume ratio? Well, here on this slide you can see the separation of 3 different platforms, the porous 5 micron, the solid core 2.5 micron, and without correction for VD over VM, and a solid core 2.5 micron with correction for VD over VM. And if we align up the different peaks, we can see how correcting for VD over VM is very helpful to ensure we get the most accurate translation possible. So whilst we've talked about speed, there's also an opportunity for resolution. So I've talked a lot about how you can speed up your separations now using method translations. How about getting extra resolution? Well, here we can see between the porous to porous separation, the top chromatogram to the right chromatogram, we've increased the column length to give us extra resolution between the peak pairs highlighted. But what does that look like for a more complex sample? And here we have an echinacea sample, which is a natural product, and we can see here that we've got an ultra-core column 150 by 3 millimeters. Now we can use our 5 translation steps, and what we can actually do is add another column to the separation. So we increase the bed length. This will give us more resolution and also hopefully increase our peak capacity, which it does, as we can see on the slide. That's 2 columns daisy chained together to give high resolution. And then we can take it to 3 columns to give us peak capacity of over 900. And if I show you the detail highlighted in red there, we can see as we go from the top chromatogram to the middle and bottom chromatogram, you can see the extra detail appearing in that complex sample as we're increasing the column length, but at the same time increasing the peak capacity of the separation. So translations really can be powerful for these complex samples. So just to bring that together for translations, it is possible to do accurate translations. For isocratic translations, consider length over particle size ratio. It is the key for isocratic translations, but be aware of dispersion effects on small columns. For gradient translations, it is more complex, but armed with the right equations, it is possible to get pretty accurate translations. And consider the dwell volume to column volume ratio impact. This will help you get a more accurate translation. And with those natural product samples, you really can get high peak capacities by daisy chaining columns together to get much higher resolution. So just to finish that up now, because I'm a few minutes later than I'd hoped to be, hopefully you've heard selectivity is the key in chromatography. Understanding selectivity can be very powerful. It helps you with your method development, helps you achieve your goal faster. Screening columns with different retention mechanisms exploits selectivity and helps you achieve your method faster. From a translation perspective, using the equations I've described, it can be very straightforward for isocratic and gradient separations. You must consider extra column band broadening or dispersion effects for smaller columns, as it will reduce the quality of your chromatography. And of course, these complex samples, you can maximize your resolution using increased bed length by daisy chaining columns together, if that's what you want to do. So thank you for listening to my webinar today. I hope that's been informative. I'd like to now pass back over to Laura, who will begin the question and answer session.
Thank you very much, Alan. That was extremely informative. It is time to get started with the questions and answers, but before we do that, we have 2 brief polling questions for our audience. For all of you in the audience, you can click directly on your screen to submit your answers. Okay, so here's the first question. What is your solid core vendor preference for HPLC or UHPLC separations? Waters Cortex, Agilent PoroShell, Phenomenex Kinetics, Sapelco Ascentis Express, Thermoscientific AccuCore, AMT Halo Fused Core, or other. So once again, what is your solid core vendor preference for HPLC or UHPLC separations? Waters Cortex, Agilent PoroShell, Phenomenex Kinetics, Sapelco Ascentis Express, Thermoscientific AccuCore, AMT Halo Fused Core, or other. And you can click directly on your screen to answer. All right, let's move to our next question. What column discussed during this web seminar today interested you the most? The ASICSL 2 micron C18AR, the 2 micron C18 PFP, the 2 micron Super C18, the ultra-core solid core 2.5 micron Super C18, or the ultra-core solid core 5 micron Super C18. So again, click directly on your screen to answer. Which column discussed today during this web seminar interested you the most? And you've got that list in front of you. We'll give you one more moment to answer. Alrighty. So let's now move on to the questions. As a reminder, if you'd like to submit a question, you can do so by typing it in the Q&A box, and you can find that by clicking on the red Q&A widget at the bottom of your presentation window. Okay, first question, Alan. When you talk about the ACE-PFP, you mentioned shape selectivity. What do you mean, chiral?
Okay, thank you for the question. So no, I definitely did not mean chiral as a mechanism, stereoisomer type interactions. What I meant with shape selectivity is that depending on the characterization protocol that you use to understand your different columns and their bonded phases, there is the protocol that I use, that we use at our company, is based on what's called the Tanaka protocol, and this uses a mixture of triphenylene and O-terphenol, which give you an indicator of value for what's termed shape selectivity. And what that means is the ability of those two analytes in that mixture, in that characterization of your bonded phase, to actually interact and be retained with the stationary phase. And it's due primarily with those two analytes that I talked about that we use for characterization, due to their rotation around bonds to do with planar regions on the molecule. So if something can slip between the bonded phase, then it can interact and be retained more than if it cannot, and it elutes through the column. Shape selectivity is a recognized mechanism in characterization protocols and chromatography column characterization, and some phases have high degree of shape selectivity and some have low, and so it really does depend on the bonded phase. A standard C18 actually does have some shape selectivity in its own right, but it is definitely not chiral interactions. That is something very different.
Excellent. Thanks, Alan. What software was used to optimize temperature and gradient conditions in the method development for paracetamol?
So there are various softwares available. In the example that I showed, I think the screenshots were taken from DryLab or LC Simulator. I can't remember which one it was. They both gave method optimization modeling packages gave similar results, but I think it was either LC Simulator or DryLab. I can't remember now.
Okay, thank you. In all of your method development work, where does the existing literature come into play? Do you not first study the existing literature internally and externally to your company? And if not, why not? And how does one efficiently make use of existing data?
That's a great question, thank you. So I think existing literature is a useful resource. A lot of places that I go to do not have access to literature because mostly you have to pay for that access to different journals and so on and so forth. There are some open access databases, of course, but the really good complex literature where the popular magazines and publications such as Journal of Chromatography, Analytical Chemistry, and so on and so forth, typically you have to pay to access. So what I've tried to do here in this seminar is describe from a first principles perspective, using the concepts of chromatography to help people understand why the method is what it is and how it can be developed relatively simply, to give you the answer quite quickly based around selectivity. Obviously, if you wanted to separate aspirin and its specified from your impurities, there will be a method out there, absolutely. Mostly, the people I talk to and the problems I've had in my career in chromatography are to do with samples that there aren't methods available for, hence the need to do some type of method development. And the purpose of this is to try and help people to understand that selectivity is a great way to approach your method development, as it will help you understand the principles and fundamentals behind what you're trying to achieve.
Excellent. Does USP allow one to do method translation to other particle sizes for gradient separation, or is a complete validation work needed?
Okay, so my understanding is that the gradient changes are very, the allowable gradient changes are very, very small. I do not think that you can do what the question is asking. I think the restrictions are quite tight. But for everybody still listening, I absolutely recommend that you check with whatever pharmacopoeia that you're wanting to test to, whether it's the USP, the JP, the CP, the PharmUA, the BP, whatever it is, and check with the chromatography general chapter in that particular publication. But I'm pretty sure for gradient separation there isn't much flexibility. My understanding is, talking to the guys on the committee and some of the people who make these changes, is that there are discussions about how we can try and use modern technology and modern particle platforms and for gradient analysis and monographs as well, but it's not quite there yet.
Fantastic. Well, Alan, thank you so much for your excellent presentation and for answering those questions.
Okay, thank you very much. Thank you very much for the webinar.
Excellent. Well, unfortunately, we're out of time, so I'd also like to thank all of you in the audience for participating today. And I'd like to thank our sponsor, MacMod, for making today's webcast possible. Today's webcast will be available for on-demand viewing through May of next year. You'll receive an email from LCGC alerting you when the webcast is available for replay. We invite you to forward that announcement to your colleagues who may have missed today's live event. We hope to see you all next time. Goodbye.